Christina Morales, Bio 225, May 13th, 2018, Bacterial Enumeration, Dilution, and Plate Counts. I have already thoroughly washed my hands with soap and warm water. I have on my safety goggles, my gloves, my face mask, and my apron. I've also disinfected my work surface by wiping it with 10% bleach solution. And I've also wiped it down. I have my 10% bleach solution here. I have my distilled water. I have my isopropyl rubbing alcohol, my 6 Petri dishes. I have 1 pipette. I have 6 test tubes, test tube clamp, test tube cleaning brush, test tube rack, and my sixth pre-poured agar plates. And I also have my Saccharomyces cerevisiae yeast solution. So when I prepared my Saccharomyces cerevisiae, I used 1/4 teaspoon of Saccharomyces cerevisiae from the yeast packet into my disposable cup, and I've added approximately 2 tablespoons of warm water into the yeast, and then I swirled it until it dissolved. And then I allowed it to sit for 10 minutes until it began to froth. For the labeling, I had already labeled my 6 test tubes from 10-1 to 10-6, and I've also labeled my 6 nutrient agar plates from 10-1 to 10-6 as well. I'm also going to sterilize my pipette by grabbing a small amount of alcohol into the bulb. And I'm going to put the liquid into my disposable liquids. I'm shaking it all out to make sure that it's dry and ready. And now all the alcohol has exited the pipette. Now I'm going to add 2.25 milliliters of my distilled water into each test tube. For the sake of time, I have already added my distilled water into the remaining 5 test tubes. So now I'm going to swirl the yeast solution to disperse of the organisms evenly. And now, with my pipette, I'm going to grab 0.25 milliliters of the cerevisiae solution into the test tube labeled 10-1. And then I'm going to rinse my pipette with my distilled water in between each transfer to make sure it is clean. I'm going to continue this process for the remaining 5. I'm going to then transfer from 10-1 into the 10-2 test tube. 0.25 milliliters. And then again rinsing it with my distilled water to make sure that it is clean for the following procedure. Then I'm going to get 0.25 milliliters from 10-2 and transfer it into 10-3. water it down with my distilled water to clean it, and dispose of it. I'm going to continue this process for the remaining. And again, cleaning my pipette. I have now rinsed my pipette in my distilled water to remove all organisms inside and out. Now I'm going to pipette 0.125 milliliters, which is approximately 4 drops, of the Cerevisiae solution in the test tube 10-1 onto the agar surface of the corresponding plate. So I'm going to take the lid off. Grab test tube 10-1. I'm going to get 0.0125 milliliters and place it over the agar. I'm going to put the remaining in my disposable liquid. I'm going to put the lid over the agar plate and gently swirl it so that I can spread the solution over the entire surface. And then I'm going to rinse my pipette in the distilled water to remove all organisms inside and out to make it ready for the next procedure. Now again, I'm going to grab test tube 10-2 and put the 10-2 onto the agar plate, again 0.125 milliliters. And put the remaining in the disposable liquid. Cover it and then swirl it so that it gets all over the surface. I'm going to continue this process for the remaining ones, remembering to clean out my pipette. I'm now going to grab 10-3 and do the same process for it. Put the remaining in the disposable liquid. Making sure it goes all over the surface. And cleaning out my pipette. Now on to 10-4. -5. And then 10-6. I'm now going to dispose of the remaining cerevisiae solutions in the cup and test tubes by pouring them down the drain. I'm going to clean the test tubes with soap and water and return them to my kit with the test tube rack. I'm also going to sterilize my graduated pipette with alcohol and then return it back to my kit. I'm going to wipe down my work area with a 10% bleach solution. I'm now going to place my agar plate in my incubation environment. I'm going to place them there for 12 hours, and then after 12 hours I'm going to invert them, and then I'm going to wait for 36 hours. I'm going to go ahead and close my closet doors so that they are away from any light and air that may disturb the incubation process. CMorales, Bio 225, May 16, 2018. After 63 hours of my total incubation time, I checked my plates for the presence of Saccharomyces cerevisiae. In 10-1 and 10-2, my colonies of each plate are more than 300 and they are considered too numerous to count. In 10-3 and 10-4, my colonies have no more than 300 and they're considered viable. In 10-5 and 10-6, they are considered fewer than 30 colonies and they are considered too few to count. Although they're not very easy to be visible in my video, they come out more visible in my lab report.